The past 2 years have been a living hell. Almost everyone has experienced the disastrous and devastating effects of COVID 19. The coronavirus has taken the world by storm and is the root cause of all the problems in the world today. But we aim to find a cure for this deadly disease, and that is through CRISPR.
Okay, but what actually is this CRISPR? I know, it sounds very complicated, but it's actually pretty easy. So, the most used technique in CRISPR, which by now we know is clustered regularly interspaced short palindromic repeats, is genome editing. See, in gene editing, we use different types of Cas proteins, Cas9 being the most popular one. So basically, what we do is that we inject Cas9 protein along with the guide RNA in the body of the person. See, then it matches and binds with the desired DNA sequence and cuts it. Now, when this happens, some mutations are introduced, and that is what disables the.
Now, to understand that better, we can consider our DNA to be the various types of plants in our home. Our genes are stored in chromosomes that are like these different plants, not necessarily in structure, but the different roles that they play in the body. So, for example, I don't like a certain plant in my house. I'll simply take it and put it somewhere outside so that it doesn't affect the ambience of my home. Similarly, we can just cut or disable parts of our DNA so as to reduce the impact that they have
So,
on
our
the body.
approach with the experiment is to disable that particular part of the gene on which the coronavirus attacks. For example, in this particular case, we disable region on chromosome 3 called 3p21.31. It's associated with the acute respiratory failures. Once this gene is cut, it won't interact with the culprit ACE2 protein of the virus. So, of course, the person will not be infected by the disease and would develop immunity against it. The three of us had a very enriching experience throughout the whole project, and we couldn't have done this without the support of Deepa ma'am and Prashant sir.
We couldn't perform the experiment in real life as it needs to be performed on actual people, and we don't have the required equipment in our school lab. But we'd love to test our hypothesis and perform the actual experiment in real life. That was it from our side, and we hope that our efforts would be considered worthwhile.